All right, welcome to the female cycles. We're going to talk in order of the uterine cycle, then oogenesis, then the ovarian cycle. And I want you all to pay close attention to oogenesis, which is going to be similar to spermatogenesis but different, and you'll need to be able to discern the differences. So let's go ahead and start with the uterine cycle. So the uterine cycle is what's happening to the uterus during the 28 days. It's on average about 28 days. This is on average for all females. So it could be a little longer, could be a little shorter. Of course, it's going to be different pre and post, or during pregnancy and after pregnancy. It could be different in puberty, it could be different in adolescence, it could be different in young adulthood, and so on. But the average is 28 days. So the cycle starts on day one, and day one is the shedding. So day one is the actual menses, or the loss of the uterine lining, the endometrium. The first menses is what we call menarche, and menarche is important because this usually is documented in pediatrics for healthy development for females. And it's really important because it usually is where different stages of development also are surrounded by. So usually moms are responsible for paying attention to, unfortunately, moms are responsible for paying attention to when the first menses is. But fathers should also be involved in this process as well. Menarche usually in America is around between ages 11 and 12. That's the first one. I think the average age for America, the last time I checked, was like 12.43 or something like that. But the range of the ages that it can happen is between 5 and 16. Yeah, 5 years old and 16 years old. So what is up with that? So between 5 and 11, there has been some, I don't want to say strange occurrences, but out of the normal circumstances, where children with certain genes, they've been able to target it to a certain gene. I think it's called the MRY or MRN gene. I forget what it's called. But this gets turned on a lot sooner in children than it normally should, which triggers early menarche or even early puberty in boys. This is the same for both boys and girls, the range of being between 5 and 16 years old. I know your book does not say this, so this is something that I'm adding in here, but it's important for us to understand because there are a lot of environmental aspects and a lot of socioeconomic aspects to this, and this is really prevalent in the United States and something that is relatively being well studied, but not enough for us to have full answers for yet. There's some arguments that society has kind of pushed this age a little sooner with like a hypersexual type of mainstream media. Some arguments for environmental reasons anyway have been for food and water and certain hormones that might be in our daily interactions. But scientists are still trying to figure out the reason for why those genes get turned on a little sooner rather than later. Okay, anyway, so what we really need to know for this course is the average age is around 12. This is also the same age of puberty for boys, is around 12. Okay, so uterine cycle. There's 3 phases. First is menstrual, next is proliferative, and last is secretory. So the menstrual phase, this is the actual shedding of the lining, and we can actually, let me zoom in here on the left. So the first picture here, A is the menstrual phase, B is proliferative, C is secretory. And it's showing you where in the uterus we're paying attention to this. So in the endometrium, the functional layer of the endometrium is going to be shed during the menstrual phase. We can pair the menstrual phase to the follicular phase of the ovarian cycle. So we'll talk about that. So that's why I have follicular on both of these next to menstrual and proliferative. The proliferative phase in picture B, we're going to see a lot of new mucus glands being produced. We're going to try to rebuild up the endometrium, try to rebuild up that functional layer. So we're going to have a lot of mucus secreting glands there to provide a lot of nutritional support in case a new potential oocyte or zygote gets implanted there. So that's the proliferative phase, we're growing. Then in the secretory phase, this can be paired with the luteal phase or the second half of the ovarian cycle. The functional layer here, we don't only have uterine glands, but we can actually see the full-on ducts. So that's how we know we have a really thick endometrium, thick endometrial lining, a thick functional layer anyway, and that it's about ready to go through menses, go back into the menstrual phase. So here we have some pictures depicting what's going on. For the next couple slides, you can see in the top right-hand corner, I took this cropped picture and I put a box around what each part we're talking about. I'll show you the big picture. It's one of the summary pictures of your book towards the very end. So here we're talking about the menstrual phase. We have the shedding of the endometrial functional layer. This is menses. It happens in patches because these spiral arteries that are supplying the endometrium with blood gradually and not even necessarily systemically, but somewhat systemically get cinched off. They vasoconstrict, they stop providing blood to that area, and then that tissue has no reason to grow anymore, and so it separates itself and removes itself. Then we move into the proliferative phase, and here we have a lot of uterine glands. We're trying to restore the functional layer. We have a lot of estrogens that are helping promote this, which you can see in one of our last graphs. And then last but not least, we have our secretory phase. This is right after right after ovulation, and it's the perfect function, hopefully the perfect functional layer that's ready to potentially take on a zygote and potentially develop it into an embryo. That's the idea, but if an embryo doesn't implant, then this lining gets shed and gets rebuilt every 28 days. Lots of mucus secretions in this area too. Any lady can tell you that, or I should say not any lady, but anyone with a uterus can tell you that. Let's see, I don't think I want to tell you anything else here. Okay, so now oogenesis. So oogenesis is analogous to spermatogenesis, except there's some differences. So oogenesis begins before birth. It accelerates during puberty and it ends at menopause, versus males, they start at puberty and continue on until death. So what happens in oogenesis before birth? We talked about this in the very first video, but oogonia, those stem cells, go through mitosis a couple times to get a bunch of primary oocytes. And then those primary oocytes, we have about 7 million. And then by birth, you lose a couple million, so then you end up with 2 million. And then by the time you actually hit puberty, you've lost again quite a bit. And now you're left with about a quarter of those, so you have about 400,000 left once you actually start puberty. That's assuming that the average puberty is at 12 years old. And all of those primary oocytes, they started meiosis barely. They just touched prophase 1. So they all have the right amount of genetic material and all the right amount of cell material that they need in order to separate themselves into other oocytes. And that's it. That's all that they're ready to do. Once the oocyte finishes prophase 1, then they go through all the way through to metaphase 2, and then they stop at metaphase 2. They get ovulated, so they leave the ovary, and then they wait until the sperm. And then it's not until the sperm actually comes that they finish doing meiosis 2. The result we have after meiosis is you only have one mature oocyte, and then you don't have any other oocytes. You're going to end up with 2 to 3 polar bodies, depending on what happened to the other cells, which we'll talk about here stepwise. So what is a polar body? A polar body is essentially a cell that took off all of the extra cytoplasm from the original oocyte. And this makes a lot of sense as we talk about it, but I'm gonna come back to this point after we've actually talked about what oogenesis looks like. So this is kind of a summary slide. Let's go ahead and draw it out over here. I'm gonna zoom in closer to the right-hand side, but you can follow along with this picture on the left. So let's go ahead and start with our oogonium. And remember, this is our stem cell. Our stem cell that's our gamete, and they went through a couple rounds of mitosis. And after a couple rounds of mitosis, then we have primary oocytes. And these primary oocytes are ready to go through meiosis 1. Meiosis 1. And actually, let me break this apart even further so that we can really get a good grasp of this. So they go through prophase 1. And after prophase 1, they wait until puberty. That magical time. And at puberty, then we finish meiosis 1 to become a secondary. Let me make sure that that's correct, right? Yep, we finish meiosis 1 in order to become a secondary oocyte. Okay, so we make our secondary oocyte. Now, our secondary oocyte it needs to go through meiosis 2, but it's not going to go through all of meiosis 2. It'll go through metaphase 2. So we still have a secondary oocyte, and then the secondary oocyte gets ovulated, so released from the ovary. Then the secondary oocyte is going to wait around, wait for sperm, and the sperm will help trigger finishing meiosis 2. And then we finally have a mature oocyte, and then that mature oocyte can turn into a zygote. Let's say implantable zygote. Okay, oh my gosh, wow, what was that? Let's go ahead and say that again from the beginning. So, oogenesis, we start with an oogonium. These are our stem cells, those gametes. They go through a bunch of rounds of mitosis, so we have a bunch of them. We talked about all of the numbers. Until puberty, we have primary oocytes that stopped at prophase 1. So they have the right amount of genetic material, they have the right amount of cell material, but they haven't divided themselves yet. So then we finish meiosis 1 and we become secondary oocytes. And those secondary oocytes will finish going through metaphase 2. And they stop at metaphase 2 when they get ovulated. They leave the ovary. And then they're waiting for the sperm. And when the sperm come, remember the sperm are going to also have half of that DNA. They're going to be haploid. These are also haploid. And then they combine together and we finish out meiosis 2 with just the oocyte. And then we finally have a mature oocyte, and hopefully that zygote now can become potentially implantable and turn into a viable embryo. So now let's talk a little bit about what does that look like in pictures-wise. So if you remember, we went through spermatogenesis, like this, where we talked about 2N to 2N to 2N and so on. So we're going to do that now. We already talked about mitosis, meiosis, or meiosis 1 and meiosis 2. And we're not going to really talk about where this happens yet because that'll make a little bit more sense whenever we see the rest of this slideshow. So let's go ahead and, where do I want to draw this? I want to draw this here. Okay, so let's go ahead and talk about, maybe we can do it up here. I'm sorry. And I can do it in a thinner, different color. Okay, so here we have a 2N, right? And we're going to go through multiple rounds of mitosis. And we're going to do this a couple times, right? So we're going to have a bunch of these and so on, right? Then they stop in prophase 1. So we're going to, again, have 2N. Oh, goodness. 2N. So then again, we're going to have these 2N cells. They're going to stop at prophase 1. They're paused. They're arrested. We call that arrested in prophase 1. Let's say... Actually, let's just take one so that it's not confusing. So let's just take one cell, one oogonia, one primary oocyte. Let's take that one primary oocyte and finish meiosis 1. So just like spermatogenesis, remember, we have to divide this cell not only once during meiosis 1, but we have to divide it again during meiosis 2 because remember, spermatids, we end up with 4. So we have to do something equivalent here. So let's take that 2N, and let's go ahead and go through meiosis 1 and finish meiosis 1. So now we're going to end up with, again, another set of 2N cells. But this is kind of a special case because the oocyte doesn't want to be inefficient at all. It is so not interested in that. So what it does and it also has to be a much bigger cell than the sperm. And in order for it to do that, it's going to have to gather a lot more materials than what the sperm has to do. And so in order to do that, it's going to take over everything that was taken from it. It's going to take over all of the good stuff and basically get rid of everything that's unnecessary. So this 2N business is kind of a lie. So I'm going to go ahead and erase this 2N here. And this 2N I'm going to draw as a bigger 2N. So it's going to have a lot more cytoplasm in it. And this leftover stuff is going to have just that genetic material, but none of the cytoplasm, none of that extra like mitochondria, Golgi, bodies, all of that stuff. We're not going to have any of that. It's just going to be left over. And we're going to do this again through meiosis 2. So let's take the big 2N, the big secondary oocyte, let's take that through meiosis 2. So after meiosis 2, this again is going to divide, right? And we're going to have N and N, right? And the same principle is going to apply. So that big cell wants to hold on to all of the material as much as possible. So this N is actually going to hold on to a lot of stuff. But forget that it's ovular, draw it a circle, however you want to draw it. And then this baby N is going to have basically nothing. It's just going to have the genetic material, and it's going to put it off to the side because we don't need that genetic material. The one in the big one is much better for us. So we're going to hold on to that. So let's talk about what happens to this baby 2N. So this baby 2N, this baby diploid cell, I shouldn't call it baby. Well, they're called polar bodies. So let's label them now. So these are the polar bodies. And polar bodies are just a plasma membrane, barely, and some genetic material. That's literally it. These are just the extra amount of DNA that the oocyte didn't want. So that polar body has the potential to go through meiosis 2. It can, it might not. So this is why we get a variable 2 to 3, because this 2N could go through and separate itself into another set of polar bodies, and then we would have 3. Or it could just remain and continue on, and then we would just have 2. We would just have this one, and we would just have this one. So you have a range of having 2 to 3 polar bodies once you're done. Let's talk about what's in this one. So this one is just going to have half the genetic material. This one is still carrying, it's still a diploid cell, because remember we had tetrads over here, and so we separated those homologous chromosomes into 2 separate cells. And then let's talk about what's happening in this cell. This is a haploid cell, so it only has half the amount of genetic material. It's going to have 23 single chromatids that will hopefully match up with the sperm's sister chromatid, the matching chromatid. And we can only really get to this final mature ovum, mature oocyte, or mature ovum. Ovum would be singular, or ova would be plural. And we can only get to this if we have a sperm, and a sperm has to come, and there's all this chemical series of steps that has to occur in order for this secondary oocyte to finish meiosis 2 and become a mature cell. So we have to have that sperm in order for it to happen. If we don't have the sperm, then we just lose this in metaphase 2, and that's it. It just stops at metaphase 2, and it doesn't do anything else. It just gets shed during menses, and that's it. Nothing special. But like I said, if it goes through meiosis 2, we can lose this second polar body, potentially, leaving us that range of 2 to 3. Okay, let's continue. So that's oogenesis. And here I have because oogenesis is kind of complicated, so here I have a broken down what's happening, what, and where. So hopefully this is helpful to you. If it's not, then just ignore it and find another way of writing out the same material. But you really need to be familiar with what's going on in oogenesis and be able to compare and contrast it to spermatogenesis. Okay, so here I have this image that I made for you all. So in the bottom left portion of the picture, this part, I again took this and I cropped this from that ultimate big summary picture that your book gives you. And this is just giving you the ovarian cycle. It's not telling you what's happening in oogenesis, and nothing in that diagram actually does tell you what's happening in oogenesis. So I made my own oogenesis slide that pairs with what's going on in that ovarian cycle. So here, and we'll talk about what happens in the ovarian cycle, but let's just say that this is the first half of the ovarian cycle and this is the second half of the ovarian cycle. So the first half of the ovarian cycle, it actually is the ovarian cycle is only concerned with what's happening with the cells that are supporting the oocyte as the oocyte goes through oogenesis. I'll say that again. The ovarian cycle only creates and manages the supporting cells that are surrounding the oocyte as it goes through oogenesis. So whenever we think about oogenesis, we actually have to think a couple steps before the very first menses and before the very first ovarian cycle. And we have to start with that oogonium, right? Because that's what's happening in utero as the female baby is growing in utero. And then we finally make those primary oocytes, and then those primary oocytes go through meiosis 1, and then those primary oocytes go through meiosis 2. And I have the first polar body depicted here too. So this picture will hopefully make so the top portion should make sense to you. The bottom portion will make a little bit more sense whenever we talk about the ovarian cycle here up next. But let me talk about where I got these pictures from. So I took these pictures literally from, I just cropped and took all of this, but I made it linear instead of top to bottom, so that it actually lays on top of what's going on in the other processes, so that it helps paint a complete picture. So if this doesn't make sense right off the bat, that's okay. Spend some time figuring out how to tell the story in your own words. And when it comes time to matching things up with what cycle is happening what and when and how do we call that, then come back to this picture, and hopefully it'll be helpful. In the top right-hand corner, then I have what's happening on the left side is oogenesis. So a top-down picture, the exact same thing that was already pictured in your book. But what I really like about this picture is it shows you point by point where that oocyte is during the ovarian cycle, which is depicted on the right-hand side. So it's the exact same thing that's going on in the picture I made down here, right down here, except it's showing you what it looks like in the ovary. So hopefully this will be helpful for you too as we go through this. But it shows you like where the primary oocyte is and what we call that as a follicle, and where the secondary oocyte is, and it being ovulated from the ovary and it leaving. Okay, so hopefully those will be useful for you when you come back and try to put it all together. Okay, so now let's talk about the ovarian cycle. So the ovarian cycle comes in two phases, the follicular phase and the luteal phase. And the follicular phase is named the follicular phase because it is building up the follicle. And then the luteal phase is named the luteal phase because that's what's left over from the follicle. And here, again, I have the same picture. Let me zoom out on the right side. Okay, so here I have the same picture. I cropped that out, and I boxed in the follicular phase, and the right-hand side, which you'll see on the upcoming slide, is going to be the luteal phase. Excuse me. And then down here, I took the pictures from the book of what's happening during the follicular phase. So step 1, you have a primordial egg nest in the ovary. Oh my God, what does egg nest? We're not birds. So primordial. So this is like almost prehistoric. You can think of it prehistoric oocytes that also have some sort of supporting cells surrounding them. So we call these primordial ovarian follicles. Then we go into step 2. Step 2, we have a formation of a primary ovarian follicle. And the primary ovarian follicle we can identify because we start seeing granulosa cells and thecal cells. And those are going to be really important to us whenever we talk about what they do and what hormones they're releasing. We can also tell it's a primary ovarian follicle because we see a primary oocyte in the middle. So primary oocyte, primary ovarian follicle. That's nice and easy. Okay. The zona pellucida is nice to know, but we don't talk about it in details in this course. The zona pellucida is theoretically what the sperm is supposed to penetrate through in order for it to fertilize the cell. But like I said in a previous video, that's been proven to not always be necessarily true, so we don't highlight the zona pellucida too much in this course. Okay. If you're interested in the zona pellucida, though, they talk about it in chapter 29, so if you're interested. Okay, then step 3. Now we have that primary ovarian follicle. It turns into a secondary ovarian follicle, and we can tell it's a secondary ovarian follicle because we can see the definition of those granulosa cells really thick around that primary oocyte, and those thecal cells are around as well. But this is kind of confusing, right? Because now we went from a primary ovarian follicle that had a primary oocyte to a secondary ovarian follicle that still has a primary oocyte. Is that really true? Yes. So remember, because we go from primary oocyte to primary oocyte, and then we go to secondary oocyte. So right here, we have primary ovarian follicle with a primary oocyte, a secondary ovarian follicle, again with a primary oocyte. And then finally, we had the tertiary ovarian follicle, step 4, and this is where we finally have that secondary oocyte. And we can tell it's a secondary oocyte because we have the corona radiata surrounding it. We have this huge space filled with a bunch of cytoplasm. We call that the antrum. The granulosa cells are still there. You can't see the thecal cells in this image, but they're still there. And the secondary oocyte is what's going to be ovulated. So this is the end of the follicular phase, is all of these 4 steps. We built up to the tertiary ovarian follicle, sometimes called a Graafian. This is an old name. Graafian follicle is the tertiary name, or the tertiary follicle. That's an old term. We don't use that anymore, but if you're Googling things, it's the same thing. Now I want us to focus on the top right-hand picture. So this is the same picture that comes from your book. And this is showing you an ovary. And it's showing you generally where all of these steps are taking place, but that's a total, it's just kind of a crapshoot of where this is happening. But we do know it's always happening in the cortex. So let's start with the primordial ovarian follicles. The primordial ovarian follicles give rise to the primary ovarian follicle. The primary ovarian follicle is going to have a primary oocyte in it. Then that primary ovarian follicle is going to continue to develop to become a secondary ovarian follicle. That secondary ovarian follicle has a primary oocyte in it. Then that secondary ovarian follicle turns into a tertiary ovarian follicle. That tertiary ovarian follicle has a secondary oocyte. And then that secondary oocyte is what actually explodes out of the ovary. We call that ovulation. That's when the secondary oocyte is released. So the corona radiata comes with that oocyte, and the corona radiata is going to be a supportive set of cells surrounding the secondary oocyte, releasing a little bit of hormones, telling the oocyte where to go, and supporting the oocyte as it travels down the the uterine tubes, and implants onto the uterus potentially. Okay, so what happens to all the rest of those cells that are left behind? We can see that little bit of purple cauliflower-looking structure left behind. Those cells have an extremely important purpose. They stay behind and they tell the body, hey, I released an egg. You might get pregnant. Let's hold down the fort and prepare in case you do get pregnant. And let's just pretend like you're pregnant for now, so then that way we don't get rid of any potential good eggs, right? We don't want to let go. We don't want to induce menses too quickly because we want to make sure that there's enough time for a potential sperm to come and fertilize that egg. Okay, so those remaining cells stay behind in the ovary, and they turn into what we call the corpus luteum, that yellow big flowery structure there. Corpus meaning body, luteum meaning yellow because it literally looks like a yellow body under a microscope. So that corpus luteum is really important because it's going to start secreting a hormone called progesterone. And progesterone is telling the body, hey, let's pretend you're pregnant. You're not pregnant, but let's pretend you're pregnant. And this is going to prevent you from going through the menstrual phase of the uterine cycle. So then that way we can leave that secondary oocyte in place, potentially waiting for a sperm to come and fertilize it. Then after a while, right, corpus luteum may or may not need to be there anymore. Maybe that oocyte gets fertilized, maybe it doesn't. Let's say it doesn't get fertilized. So no sperm, no fertilization, no baby. We go through menses. That corpus luteum doesn't want to continue secreting progesterone anymore because we want to have another ovarian cycle. We want to release another secondary oocyte. And so in order to do that, that corpus luteum will regress and actually turn into what we call the corpus albicans. Albicans meaning white, so white body, essentially scar tissue. And then that doesn't secrete anything. It just hangs back and that's it. That just becomes part of the ovary now. Typically, these ovarian follicles sit around the cortex, and the cortex is where these are being developed. Then by the time you get to the corpus luteum, corpus albicans stage, then you're finally in the medulla of the ovary. And this is why whenever you look at a ovary of an older woman, maybe someone who has had children even, you can actually feel these scar tissue balls almost in the ovary. And that's just the actual scar tissue left behind from the corpus albicans. Okay, so that all that we just talked about. I got excited. I got a little ahead of myself. So up until the tertiary ovarian follicle is the follicular phase of the ovarian cycle. Here we have microscopic pictures of what's going on during the luteal phase, corpus luteum, luteal phase, where we develop the corpus luteum and then eventually turn into the corpus albicans. So step 5, you ovulate, this is an awesome freaking picture of the secondary oocyte leaving the ovary. Crazy. It's just super cool. And then step 6, you actually have the corpus luteum. Terrible picture because it's red, and I just told you that it's yellow, so I promise it's yellow. And then step 4, the formation of the corpus albicans actually looking white. How exciting. This picture in your book doesn't show it in this order. It shows it like step 1 to step 4 to step 5 and then to 6 and 7, which I think is a little ridiculous, so I just reordered them. So please don't be confused. I just cropped them and put them in order here on the slide. It's just trying to show you like the cyclic event that happens in the ovary. That's why I think they made it like that. Okay, so follicular phase up until tertiary ovarian follicle, luteal phase, you have the corpus luteum and then potentially the corpus albicans, and then you start all over again, right? So this is the ovarian cycle, or the ovarian cycle, where we are nurturing and taking care of the oocyte as it goes through oogenesis and eventually becomes ovulated. All right, so here we have this all written out. So like I said, tertiary follicle, also called I misspelled Graphian, perfect. On here I misspelled Graphian, it's correct on the slide. The 2 phases, right? Follicular phase is the first half of those 28-day cycles, so the first 14 days. By the time you get to about day 10, day 12, the tertiary follicle is about ready. By the time you have enough FSH, then you have ovulation, which will show this in a graphical representation. The antrum and the remaining surrounding cells stay behind, turn into corpus luteum. The ovulated secondary oocyte will be released with the corona radiata to support it. And then follicles that do not get released. So let's go back to this picture. So here we started with primordial ovarian follicles, and you see how there's like 1, 2, 3, 4, 5, 6, 7, 8 of those. Oh goodness. So not all of those actually went through this whole process. This picture shows you one going through this process. It's actually about 3 to 5 per cycle going through this process, but not all of them actually get ovulated. Only one will get ovulated. This is a really cool biological feature of a timing thing, and I forget what part of the brain does this now, but it will literally timestamp when each oocyte is ready to go, and the one that lines up with the right timing is the only one to actually leave. But the other 3 could have potentially left, but just because of timing, they just didn't meet the cutoff, so they don't get ovulated. So they just turn into corpus luteum, just like the other remaining follicle cells do too. Okay, so we call that, the ones that don't get ovulated, we call that follicular atresia. Okay, so that is the follicular phase. Let's go into the luteal phase. So the luteal phase, we're only talking about the corpus luteum here. Corpus luteum starts to form with the help of LH, starts secreting progesterone. Progesterone tells the body, hey, let's pretend that you're pregnant for a little bit. Let's not get rid of anything. Let's prepare the uterus for potential implantation. And then if there's no implantation, then it regresses and turns into corpus albicans, and progesterone and estrogen levels drop. Okay, so back to this slide. So here we talk about the thecal cells and how the thecal cells are releasing estrogens. We're going to talk a little bit more about that here in the upcoming set of slides. So this is it for the female 3 cycles. We started with uterine, then we progressed into oogenesis, and then we talked about the ovarian cycle. But remember, all 3 of these are happening at the same time. So please understand what's happening during each of the 3 before we put it all together.